to Science Track panel. How do drugs get made? Yes, that's... Oh my God, sorry, I blanked out there. Just a few quick announcements. DragonCon's charity is called Open Hand. Their mission is to make sure nutrient-dense food gets to those who are in most need, particularly those folks who are in food deserts, so elderly and those who cannot access good nutrition. So, and please make DragonCon pay. They match up to 100,000. So please, please. You will also have noticed that I've taken my mask off. That is for folks who need to be able to lip read to see. I will take my mask off and on, but I need you all, please kindly keep your masks on. And so with that, I will introduce myself. My name is Mouse Hecht. I am the moderator for this panel. I am a elementary school teacher, and we'll just go down the line for everyone else to introduce themselves. Thank you.
Hi, I'm Liz de Colvenaire. I am a computational chemist. I work in computational drug discovery, which is a little bit less of how we discover drugs today and a little more of hopefully how we discover drugs tomorrow.
Hi, I'm Dr. Jen Hardy. I work in a biotech company out in the San Francisco Bay Area. We are working on a CAR T therapy for cancer, and we are in phase 1 clinical trials.
Hey, I'm Jess Eddington. I'm a clinical research associate, which just boils down to being a science cop, and we are not all bastards. But I got an MPA and did my paper on Black inclusive policies in clinical research. I have a bachelor's degree in psychology. I worked in clinical research for about 5 years.
My name is Katie Cross. I'm currently a young adult fantasy author, but I was a pediatric and cardiac registered nurse before I got into writing. I have my bachelor's in nursing. My mother-in-law is a hospice nurse and a nurse educator, and my aunt is she's a pediatric nurse practitioner and runs her own business. So I'm here to give the nursing side of drug life.
Hi, my name is Jessica Kale. I am a neuroscientist, and I teach statistics and research methodology at Pepperdine University in Malibu, which is nice and cushy. And I am also a scientific consultant for movies and television and a science communicator.
Excellent. So sort of my first question is, so what has sort of been the history of drug and or like vaccine development throughout even old times to now? Like, what's sort of the history of that?
So I feel like things were fairly loosey-goosey for a long time. The sort of what we think of as the modern FDA and the modern process got kicked off in, I don't know, about the 70s. And a big part of that was due to thalidomide, which you guys might be familiar with. This was a drug designed to prevent nausea. They said, great, let's give it to pregnant women. It caused horrible birth defects. We didn't have a lot of that in the US because there was one woman at the FDA who held the line and said, I don't like this data. I want to see more results. I'm not going to approve this. She came under incredible amounts of pressure, but held firm on that. So most of the problems with thalidomide actually showed up in Europe. And that, I think, was the first big crack in the system in saying, okay, no, this is really important that we do this right, and that the FDA does have broad powers, and they can't just be hand-waved for people that throw enough money at the problem. So I think that was sort of the crystallizing moment for how our system works as it does today.
So that is true in regards to vulnerable populations, which include pregnant women, prisoners, elderly. It really, a lot of our regulations started with the Nazis, unfortunately. There is a document called the Declaration of Helsinki that came out after the horrible practices of Nazi doctors. And that is really where the FDA kind of began and saw the roots in improving those practices.
All right, so that's sort of the history. So at least now, how do we go from I have an idea? How do we take that idea and then get it out to the public, depending on how good the science is?
Okay, I'm going to talk a lot on this panel. Sorry, guys. So you begin with an idea, and you test it in rodents, usually, small mammals. Sometimes it starts in vitro, actually. That's becoming much more, in vitro. That's becoming much more common. And then you once you see positive results from that, you can move it into preclinical studies, which is a couple of patients, usually at a single institution. And what you're looking for is not really efficacy at this point. You're just making sure that it is not actively killing people, which is really important. And then you can move on to your phase 1 studies, which is where you're taking your adverse event. I'm so sorry, guys. I did not sleep a lot last night, so I'm going to blank out every once in a while. But you're going to do your phase 1 studies where you're collecting your adverse events and, again, making sure that your drug is not actively killing anybody. And then you move on to your phase 2 and 3 where you're broadening your population scope and actually checking for efficacy at that point. And hopefully, if it works better than the current standard of care, you can take it to market and you go to phase 4, which I'm honestly not super familiar with. That's where you're doing marketing and the not fun stuff anymore.
Yeah, from my perspective on the actual drug company side of things, we spend a lot of time doing lab research on this stuff. And most things don't pan out. I think this is something people don't realize about pharma is I think we got most of the low-hanging fruit already. The stuff that's left are very hard nuts to crack. So most companies, they've got great ideas and maybe they work in mice, but as soon as you try scaling it up, turns out mice aren't people. It doesn't always translate, especially things like Alzheimer's drugs have a lousy track record. We just don't have good model systems for a lot of conditions we'd like to study. Like, how do you study the complex issues of diabetes in something that lives in a cage and in an extremely controlled environment? So there's a big disconnect between what works when you're testing it on a bench in a petri dish or in a lab mouse. We call this the mouse house, where they all live. And then when you get into people, it fizzles out a lot of times. And the first thing, as she was saying, you got to prove it's safe. Who cares if it works? We want to know if it's safe. If it's not safe, nothing else matters. So that's sort of step 1, is proving that this is okay to put into a person. And you're also trying to figure out dosing level, because, okay, you've got a drug, great, but what's the right dose to get it to work but not have too many negative side effects? So there's a lot of that going on at the beginning. And then once you figure out, okay, this is sort of the dosing space we're in, and we've shown that it's pretty safe, all right, we're going to try that in our next phase of our clinical trial. We're going to try it in a few more people at these very specific dosages that we've decided on. Okay, how did that look? And then you start looking at, did it work? So now you've proved that it's safe. Now we look at, does it work? And you're doing that in a smallish group of people, 40 people?
You mean phase 2?
Yeah.
Yeah, about 40.
So yeah, like first phase is like a dozen people. And then
They usually use what's called a 3x3 model. So they do 3 in one dose, 3 in the next dose, 3 in the next dose, and then evaluate from there. So it's a really small group.
yeah, so that's sort of, is it safe? And then you go to does it work, and you get up to more like 3 or 4 dozen people. And that's where you're testing, okay, how's this working? Which dosages are showing that they do the job? And once you've decided, okay, this is working, this is our dose, we're happy with that, then you move into phase 3, which is we try this in a whole lot of people. Because then you're playing with statistics of large numbers. You want to find the rare, weird side effects that only show up if you try it in 800 people, but didn't show up in your little group of 40. So when we were doing the vaccines for COVID, they were trying in like 40,000 people, which is huge, because they were trying to catch every little possible edge case. If you're going to give this drug to everyone on the planet, you need to know what the 1% event is. So that's sort of the
And just to give context to how exceptional that is, usually phase 3 is a couple of thousand.
Yeah, so that was extremely unusual. But they needed to do it because of how many people were going to be getting it.
So I have a question there. So when you're starting to expand, how many people you're pulling into this, is that where, are you finding these people through medical clinics? Do you kind of have other funnels that bring the people to you that would be participants for that drug? Because I've worked with doctors' offices before. It's like we try to get people to sign up for specific clinical studies. I'm curious, at what point in your process are you coming to doctors' offices and saying, hey, we need people that fit this criteria to test this drug, and then get their signature kind of thing?
Would you mind taking your mask off, please?
Oh, sorry.
I think I'm just going to leave mine down. It's too hard doing
Yeah.
upsy downsies.
I'll rephrase that. I've worked in doctors' offices where we will have companies come to us and ask us to find patients that fit specific requirements so that they can go into clinical studies at a certain phase to test whether the medicine is doing what they need it to do. And I'm curious where that is in like the phasing for the company.
For my company specifically, we are working with some of the big cancer institutes, so like MD Anderson, Fred Hutchinson, places like that. And they have doctors that work with clinical trials all the time. So there's sort of these expert centers on working with clinical
We're going to have to
trials.
check later. I might have one of your drugs.
You might. So you start off with these sort of like these centers that are used to doing this work, and they bring in their sickest patients.
Yeah.
It's it's not ethical to test a new drug in somebody that has other options. So what you're getting is people that have tried everything on the market and it hasn't worked. So you're getting very, very sick people that have been cleared, and it's usually a really small number of very, very sick people. So your early results are not necessarily all that indicative of what's going to happen when you get it in somebody earlier in their illness progression. So that's always a fun thing to try to disentangle. So the early stuff might not look as good as it really will once you roll it out, because you are dealing with people that are extremely ill.
Okay, yeah, that makes sense.
I want to clarify that the extremely ill population that she's referring to is for the phase 1 specifically. Usually when it's rolling out to healthier patients, it's around phase 2 to phase 3, because you do want to see, is it going to be effective as a first-line treatment? Because potentially this is the replacement for what you're doing already.
Which fits with what I've seen as a nurse. We would have like specialists come into the hospital for a specific thing, and some of our most severe kids or the heart people that I worked with, rarely they'd be like, they really qualify for a specific clinical trial that we're trying to help out, but usually it was that rare case, like more advanced, versus other times we had people basically handing out flyers, like we're trying to do this clinical trial for this specific ointment or something, and almost anyone could enroll. So it kind of varies when you're talking about getting it from the company into the people, from what I've seen.
They're always doing a cost-benefit analysis on ethics too, and so if you're going to be one of the first people in a trial for, an early phase, if there is any potential risk of symptoms or side effects or whatever like that, you'd give it to somebody who probably would be more likely to benefit, and the risk wouldn't be so high. But at the point where you're starting to talk about putting it into everybody, you ought to be further along and know that the benefits outweigh the risks, even for somebody who doesn't have symptoms.
I wanted to roll back a little bit to something that you said earlier about low-hanging fruit, and that's, I think, another thing about drug development that's worth looking at. So how many of you are familiar with Moore's law? The number of transistors, in a chip doubles, like, I think every 10 years is it. So in drug discovery, you get something called Irum's law, which is basically that the amount of money you have to spend to discover each new drug is also increasing exponentially. It's rather ugly, and safety is really, really, really important. It's really, really expensive, I think, as you all hinted at, to do these large human trials, even to do, a few people. So one of the things that we're hoping to do in computational drug discovery land is we can't necessarily work out the safety ahead of time, because it's really hard to tell, okay, I'm going to put this molecule into someone's body, and there's hundreds of millions of places it could interact weirdly and go wrong. But if we can use computer models to first figure out, will this molecule even treat the disease we're trying to treat to begin with, we're hoping we can, instead of requiring that you look at, say, 1,000 drugs to get each new useful candidate that makes it past phase 1, we can maybe get that down to 100 candidates, to get one that makes it past phase 1. And that's something that I think is really exciting for the future, is how we can take computer modeling of the things we can understand and help that with the rest of you when it comes to parts that are harder to understand, which is usually where the drug maybe isn't working quite right or is doing something harmful.
So my curiosity was piqued when you talked about sort of the low-hanging fruit. Could you give me examples of drugs that were just easier to discover sort of back in the day?
I mean, a lot of the early stuff was taking folk remedies and turning them into real pharmaceuticals. You know, we've known for hundreds of years that willow bark is good for headaches, but it took somebody in Germany at Bayer sitting down and figuring out how to turn it into aspirin in order to make sure it stayed in the body long enough to be useful and didn't give you a horrible upset stomach. You know, so there's a lot of that kind of thing that was done early on. And finding, we call these small molecule drugs. If it's a chemical, you know, it's a little structure you could draw with rings, then those tend to be the ones we found first.
Smolecules.
Molecules, yes. Whereas these days, more what you are probably seeing in the news are biologics. Those are proteins, they're antibodies, they're things that are made in an organism, not in a big synthesis tank. And that's because we run out of a lot of things that you can make in the big synthesis tank that will do the job. And biologics are harder to make, and so unfortunately they're also much more expensive to make. Then you end up with sort of this perverse incentive of the companies making so much money, then they want to keep studying the biologics, and maybe there are a few more small molecules left out there.
I was just going to add also, when you start moving away from small molecules, it becomes much harder to deliver them. So not only is it more expensive to make, you can't put it in a pill and take it. It'll get destroyed in your stomach. It'll get destroyed by your liver. A lot of these things need to be, say, injected. But I guess one other statement on low-hanging fruit is, you know, when you're treating diseases that a huge number of people get, it's very easy to find people who have it and run a clinical trial and figure out, you know, for efficacy. If you're looking at rare diseases, if you're looking at cancers that have already evolved as resistance to several drugs, you're already trying to treat a problem that people have tried a lot before with and failed. And B, the number of people you have to test for efficacy becomes very, very small and infrequent, which really drives up research costs.
Yeah, one of the issues that we run into is, trying to develop drugs for rarer conditions. There's not a good economy of scale there. You know, we've said, you know, drug companies have a lot of shots on goal that don't work, and they're all really expensive. So there actually are some government incentives for companies to work on diseases that don't have a lot of sufferers, but are really gnarly to treat. So there's like the Orphan Drug Act, which basically, okay, you want to develop a drug for something that doesn't have an existing treatment. It's got, you know, 800 people get it a year. You're going to be able to license that drug and have it be yours for a longer period of time, so you can make your investment back than you would for something like a cancer drug where there's a lot of people that get that disease. So there are some sort of government structural ideas trying to make sure that these rarer diseases do get addressed.
For a personal reference, my partner has cystic fibrosis. He takes a medication that is specific for his genetic deformity. Another CF patient cannot take this med, and we lovingly call them his flat screen TV pills. They were $250,000 a year without insurance. So it can show how expensive some of these more targeted drugs can be, because you put so much money into it, and then it's like, oh, well, we got 2 people who need it. So, and that's unfortunately the tragedy of our economic system, is that these drugs are so expensive, and thankfully there are acts to help fund them. Just to sort of want to pivot a little bit towards sort of when you're doing the large disbursements, kind of like with the COVID vaccines and collecting that data, and it's like, how do we ramp up stuff like that when we do have to do big deliverables of, like, say, a vaccine for a global pandemic?
So one of the really cool things about the COVID study is, instead of rolling out like a phase 1, a phase 2, and a phase 3, they bundled it. So they rolled out a single protocol and as a phase 1, 2, and 3 with the stopgaps at each transition point. And so what that means is, instead of like, you run your phase 1 trial, you get the data, you spend a year, and then you move on and develop the next phase protocol and then distribute it to all the same hospitals. By just taking that admin out, you're able to just push through it in a year. which was, I mean, incredible. And I'd love to see like what happens with that model in the future, because that is not something that has happened before.
Normally you want to pause because you just spent a lot of money, and you need to
You're
raise,
broke now.
Yeah, you need to raise more funding. You need to prove to people, hey, look at my great results from phase 1. Please give me more funding so I can run phase 2. With the COVID vaccine, everybody was funding it. There was no question that you would have the money to kick forward into phase 2 and 3, so you didn't have to wait.
And another like really good example of that in a non-pandemic year, because I'm sure you guys are tired of hearing about the pandemic, is childhood leukemia. Since the 70s, we identified that problem and we said we are going to put so much money into that, and we're going to find some really good treatments out of it. But it's one of the things that like everybody donates to childhood cancer, and when you do, you're donating to childhood leukemia most likely, because that's the most common one. And you get these great results, but just because there's just so much money out there to do amazing things.
It's important for the public to note that just because you've eliminated some of the administrative gaps between stage 1, stage 2, stage 3 doesn't mean you've shortcutted the research itself, which a lot of people, is a huge, misunderstanding. So if you streamline the process, it doesn't mean you rushed the science. It means you rushed I sent this big stack of paper to the FDA. They send it back, and this goes to this and back and forth. It's the red tape that was cut, not the science.
And even from like our perspective, we do budget negotiations with the hospitals on behalf of the pharmaceutical companies, and I've been sitting on a study since January, and we are still not open just because what were we debating? We were debating how much we would pay them to do a final phone call. So there's all this like little stuff that gums up the process, unfortunately. And I mean, that's one hospital, one institution. Like, they're doing this in such a broad scale, but if you have this nice little packet of protocols and you're not doing that every year, you already got it set and ready to go.
What we ran into last year is we were trying to kick off our phase 1, and we're trying to get submit our book to the FDA with all of our preliminary data, have a meeting with them so that we can get our phase 1 trial approved. And they basically said, we're so sorry, guys. We are completely swamped with COVID treatments right now. Like, we can't get to you right now. Like, put a pin in it. We'll get back to you in like 4 months. Just try again in January.
So having sort of this pandemic can really gum up the works, not just sort of society-wise, but even with research-based, when people are like, oh, no, we really need to put the priority on this because it's a bigger fire to put out. Katie, I know you mentioned that from the nursing standpoint, and you also are had worked with small children. When I think about drug efficacy and stuff like that, like, do the tiers of ages of humans, do they put more like concern about that? Because like I know kids' metabolisms are different from adult metabolisms, and how that works.
So in the rollout for pediatric drugs on my end, we're pretty much standing on their shoulders. When it gets to me, like, the doctors that I'm working with, they're the ones that are saying, these are the dosing parameters, these are the kids that it qualifies for. We're going to start, almost always, we're going to start here on the lowest end of the dosing parameters, and we're going to venture carefully. And some of that depended on how well known the medication already was. If we're looking at some of our cases, then, I mean, it's obviously going to be far more conservative than in children. Also, I saw that in more of a geriatric population when I worked cardiac, because everyone's liver, at that age just works differently, in their kidneys, and so you would see a lot more conservation there. With when I'd have like adults come through with heart problems, like I had a 28-year-old running a marathon that came in with rhabdomyolysis. There was a more aggressive treatment because of what's going on. So yes, I think age is a consideration in the administration of it. Also, there were situations where we couldn't administer drugs that we knew they needed because of FDA protocols. So we had a child come in. He was like 5, maybe 5. He was pretty small, and he like basically went into paralysis, and like he couldn't move his body, and we were trying to figure out why. And his family was from India originally, and after figuring it out, he was missing one of the 12, like his body could not process one of the 12 components of the vitamin B vitamin. And it's this extremely rare thing. It's so rare that we didn't have protocols for it in the US, but they did in India, and his aunt and uncle were doctors in India. So we actually they shipped it from India, and we stood in the doorway and just watched while his dad gave him the shot, and then we just monitored everything because we couldn't legally give that dose, especially to a child. So there's a greater abundance of caution when rolling these things out, and I also think there's more time given to it, to kind of talk to the families, for the doctors and nurses to be a little more one-on-one, to say, this is what we've seen, this is how it looks, this is why they qualify, and here are the implications later in life. Because when you have kids with leukemia, they may not be able to have children later with some of these drugs and that kind of thing. So you have to look at broad-term perspectives for these patients with these new drugs and ask questions and take risks they're risks that you're not going to have when you're 50 or 60.
Yeah, Europe actually gets interesting about this. They have, the European equivalent of the FDA for the EU has slightly different rules than we do here. And if you want to sell your drug in another country, you have to pay attention to what Japan's rules are and China's rules are and the EU and all this kind of thing. There are people that specialize just in particular regions because you need to know the ins and outs. They all have basically the same approach, but Europe has this really interesting extra one where they weight how much they want to fund your drug based on the age range it's meant to treat. So based on basically how many years of life are you going to give somebody if we pay for this really expensive treatment? If you are curing leukemia in a 4-year-old, that's 80 years of life you're giving somebody back. If you are treating advanced Alzheimer's in somebody who's 85, that's a few more years of life. So they actually have this interesting calculus they do, especially for extremely expensive treatments. It's based on years and quality of life afterwards.
The other thing I wanted to mention about kind of getting these new drugs past all of this and into the hands of the people is most of what we're talking about here now is those rare exceptions, those not the low-hanging fruit. But it seems like, and you guys can tell me better, there's always repetitions of the low-hanging fruit running through doctors' offices. So, I'd be working in an office and a new drug rep would come in with maybe a different hypertension med, or this statin works a little differently than this one, and they're kind of jockeying for the doctor's offices by getting the doctor's office samples and saying, well, you should start switching to diagnosing this one, even though this one has been fine. And we would even see some blood pressure drugs phasing out. So we would have people, like my grandma she started blood pressure meds in her 20s, and she took the same medicine until she was 60, and it just stopped working, and so they had to kind of switch her over. So I'm not saying there's no room for the same thing, but we also, rolling stuff out to people, we see that sort of jockeying business model happen as well.
We have an audience question out here.
Oh, wait.
Please wait until you have the mic to ask your question so everyone can hear you.
Oh, yeah. See, I'm just really loud. So kind of piggybacking off of your stuff about how the process is different in Europe or in Japan or whatever, I personally was just prescribed a medication that costs an inexcusable amount of money here, and my insurance won't pay for it because it's not approved to treat my condition here. Canada, yes. Europe, absolutely. Japan, 100%. So what steps does that take out of the process to get it approved here? If you're looking at a drug that is already approved in the United States and for sale in the United States, and I could pay cash for it, but how does that differ your study process, your stage 1, stage 2, if you want to take an existing drug for a new condition?
So it's kind of a complicated question to answer without knowing the drug and condition, and I don't want to know. Please don't tell me. But generally, what I would say about that is if they are testing it in different markets, they have generally tested it in the US as well, and the FDA generally takes a little bit longer. So it might be in the process of being approved. It might be something that they found was not marketable in the US for that, because doctors can do the off-label. And so there's not necessarily the financial incentive to do that. There's a lot of things that could come into play there. But usually when they are doing a global, which a lot of studies now are on the global scale, they are going by whichever institution is the strictest, which generally is the EU. Would you agree with that?
Yeah, I would say so.
Yeah, EU is usually the strictest, so they go off of like their parameters. So if it fits the parameters for the EU to approve it, the FDA will most likely also approve it. There are just a ton of things that can happen in the meantime that are administrative and not necessarily scientific.
Is there a place where they could find out if it's in the process? Like, the U.S. hasn't approved this? That would be kind of cool. If you could, like, track where it's at.
I have no idea on that one. You can watch clinical trials. Those are publicly available. I think it's something straightforward like clinicaltrials.gov.
It is
Yeah.
clinicaltrials.gov. I always
Uh,
put in ct.gov. It's not that. That's Connecticut, I think.
But yeah, as for where they are in the process filing with the FDA, I think a lot of the time that is very secret business knowledge, basically, whatever the technical term for that is. They don't want other companies to know what they're filing for and where they are in that process, and whether or not maybe the FDA has said, we need another data packet because that might affect their share price. And
Yeah.
so I think that tends to be more secretive.
I'm sorry.
I'll hold it. Pardon? I'll hold it. Oh, okay. I just had a question about, like, because you were talking about how they were finding new small molecules for drugs and trying to make more drugs to treat diseases. I'm a biomedical student and I've been working with cancer to try and reuse drugs, specifically antiparasitics, to treat it. How much and how often are old drugs used to try and treat new diseases?
Constantly, because it's way cheaper to take what you've already got and try to find a new use for it, right? And what we're seeing a lot in cancer right now too is like combinations of drugs. Like that's where a lot of it's happening. Really, we're looking at like new mechanisms to get it into the cell, and mostly to limit adverse events, because that's kind of the big hang up with chemo, right? Like it usually works pretty well. In some cases, it works pretty well, but it will just mess you up for the rest of your life. So they're trying to like limit that exposure. Do you have anything to add to that?
I mean, I would say, if it's an existing drug that's already been through the approval process, you've already gotten through the safety part, which is the biggest hurdle that a lot of things fall down at. You've shown it's safe in people. You have some idea what dose works. So that short circuits a lot of horsing around at the beginning. So yeah, people are always trying to find new indications for already approved drugs.
And another thing that gets nicely short-circuited is production. So especially when you're discovering new small molecules, you can draw a lot of molecules that are really, really hard to make in a lab. And there's a small group of labs that actually specialize in making new small molecules that no one has figured out how to do before. One of them is Enamine. They're located in Ukraine. They actually stopped operating for several months when Ukraine was attacked, and this was a big problem for a lot of drug discovery companies because, a lot of companies were relying on Enamine to make new compounds that they would then send out to do assays and do tests with to say, is this drug binding to the site that we care about? So when you're using something that already exists and is off the shelf, that means someone has not only figured out how to make a few micrograms of it, but someone has actually figured out how to make kilograms of it, which can be really hard when the only way we know how to do it from scratch, say, involves a 15-step process with 50% loss at each step. You can imagine then how much reagent you need to get a tiny amount of drug that hopefully is pure enough for you to do the tests you want to do.
Yeah, something else I did want to mention, I know we got some questions hanging on here, but is that there are companies, pharma companies like Pfizer have giant libraries of small molecules. They just have hundreds of thousands of compounds, either ones that have been approved or at least ones that they've cooked up and tried in other things. And whenever a new disease or condition comes along, they will actually screen a lot of those molecules against it to see if they just happen to have something already sitting there that will work. And sometimes even if it doesn't work outright, they'll be like, well, this sort of twitched in the right direction, so that's the kind of structure and the kind of drug we should go after, and you can at least sort of narrow down what you want to look at. So they have these massive libraries of these things sitting in plates, and they'll have these fancy robots that'll go pick all the ones they want to try and screen them against whatever the disease is.
So related to medications being available in other countries across the world, I'm pretty sure that in the EU, for example, pharmaceutical companies can't advertise their drugs to consumers. And I was wondering if there was any information or thoughts on if you think a similar policy will eventually make its way to the U.S., and if not, what implications you think that has.
Have you ever thought of how weird that is? It's one of the only products one of the only products advertised in media that cannot be purchased by the people watching the ad.
It's
You-
not for the patients; it's for the doctors.
Well, yeah. Well, and the doctors I've talked to said that, well, a patient will come in and say, well, but I saw this antidepressant commercial and I want that. And so, they're hoping that the brand recognition will be there. But it's a strange product to advertise it to people who can't go out and just buy it. They don't have the decisional capability on that.
In short, no. America has a really weird relationship with money and drugs. So, nah, you gotta watch those ads.
Yeah, I really wish we'd get rid of it, but I don't see it happening anytime soon. The other one that's interesting on the, Katie was talking about drug reps coming through hospitals and trying to be like, hey, I know you normally prescribe this statin, but how about prescribing our statin? If you're an extremely good-looking person, that is the job for you. They always hire these, 20-something stunning-looking people to come in and, give you business cards and free pens and all this kind of
Lunch.
stuff.
They almost
Lunch.
always brought lunch and good lunch with desserts. And a lot of them actually they recruit medical students that drop out, that decide not to go all the way through medical school. They recruit pretty heavily from that pool because they have the background in science knowledge that they need to be able to understand the drug mechanism and be able to actually talk to a doctor in an educated way about it. So if you don't really want to go be a doctor, but you went to medical school and you're beautiful and you get free lunch, go.
Oh, I just wanted to mention the clinicaltrials.gov. If anybody's interested in volunteering for a clinical study, you can sign up for alerts for different things that you might qualify for,
That's cool.
and it's really fun.
That's awesome.
Thanks. I think you touched on this a little bit earlier when you were talking about confidentiality of different applications for the FDA. I'd like to get y'all's take on intellectual property and how that's impacted, at least in the U.S., what drugs are being developed and how, and even the recurrence of changing something slightly to reapply for patents. What are y'all's takes on how intellectual property kind of plays into this whole process?
So from the IP side of things, the FDA usually has, you're good once you've developed this drug. We give you this guaranteed period of time where only you can make it, and that's really intended to help reimburse R&D costs and incentivize companies to do this work in the first place. They pay a lot of attention to the incentive structure, so it's not perfect, but they do actually think about it pretty hard and trying to make sure the right things are incentivized and the really greedy behaviors are not. One thing that companies did come up with a few years ago was, so every time you synthesize a small molecule, you get the left-handed and the right-handed versions of it, and usually only one of those is biologically active. So Zyrtec is a big 50-50 mix, only this version works. So then they came out with, once Zyrtec came off patent, then they came out with Zysol, which is just this version, but now it's a different drug because the inactive mirror image isn't in there. So that's some trick that some of the companies have pulled. I will say though, they don't always work the same way. I take Zyrtec, it works great. I take Zysol, it puts me to sleep and makes me a zombie. So there can actually be meaningful differences between those two things. But yeah, that's the major one I'm aware of that people use to try to get around the time limit.
Yeah, I guess and you see people getting around or building patent fences by, not just patenting the drug, but you do a bunch of small changes. I'm going to replace a fluorine here with another methyl and then a fluorine, because sometimes these changes can be helpful in getting the drug to, absorb better to the site, and sometimes it's just, I want to cover myself so someone else doesn't go patent this later and run laps around me in, the office free lunch routines. But that's all after you get to the finish line. So there's a huge amount of secrecy involved when you are developing a drug before you've gone into clinical trials, especially when you think you have a candidate, because it can be really easy if someone even, is seeing a presentation. If you even have a picture of the molecule up and someone takes a picture, they can work from there and steal a lot of your work if you're not very careful. So working with these things, there's a lot of internal security steps that you have to take to make sure that your intellectual property doesn't leak, before it has hit the stage where you can patent it.
And the company I'm working for, we're working on genetically engineered T cells to fight cancer, and most of that process is not, patentable. Everybody uses the same process to extract T cells from blood and to culture them and to infect them and change their genetics. Everybody does it the same way. So basically, the only secret sauce my company has is this very limited piece of the production pipeline. So usually that's why people are very secretive about that little bit. So you want to keep your piece secret, but you want also, we are scientists. We're not, the people up at the high levels, yeah, they want money. Of course they do. The people down at our level are fundamentally interested in advancing the science. We don't want to keep this stuff behind walls if we don't have to. So companies are actually pretty good, in my experience, about sharing some of these lower level protocols. It's sort of, we tried this and this is a better way to culture cells. So that kind of stuff does get passed around.
My question is about the COVID vaccine. Something that I get a lot, asked a lot, because I've taken as much of the drug as I possibly can, is people ask me, how do we know what the long-term effects are going to be, since it's a new drug?
Yes, it's a new drug. You guys feel free to jump in. I don't want to take over the panel here, but they've actually been working on mRNA vaccines for 15 years at this point. So their initial targets were obviously not COVID. They were other things like influenza, because it's really hard to manufacture the flu vaccine as it is now. It's much easier if you make it an mRNA vaccine. So we actually do have clinical trial data going back, I think, 12 years at this point on their first clinical trial that they did with this type of vaccine. So that's what we can look back on and say, okay, it's been 12 years. Do any of these people have issues that seem to stem from the technology that was used? You can't say the specific vaccine target, but you could say mRNA vaccines in general haven't had any problems that showed up within a decade and change, which I think is pretty solid.
And I guess to add to that, from the side of, okay, how does your body react to the spike, we do have data from the original SARS-CoV, the SARS disease that happened in China, at this point, 10, 15?
At
20?
least 15, 18, something like that?
Yeah, a while ago. So we have long-term outcomes on people who got that, developed the correct kind of antibodies, and survived, and we have ideas of, okay, what other effects from these antibodies, did these antibodies start causing other immune problems? And we know that, no, there's a lot of coronaviruses, and people generally don't have too many weird things from just developing reasonable immune response.
So my favorite anecdote about the COVID vaccines was, when they first came out, everybody was like, all right, this happened fast. And so I sat down to read the results of the clinical trials so that I could help my friends and family understand what was going on. For clinical trials, you have to report what we call adverse events. Every adverse event that happens to somebody in your trial, whether or not it's plausibly related to the drug. If somebody is in a car accident and they end up in the ER, that has to go in your report of something bad that happened to somebody on your clinical trial. There were very, very few adverse events in those reports, and my personal favorite was in the Moderna one. They had a patient who was struck by lightning and had to go to the ER with heart arrhythmia because she had been struck by lightning. But they have to put this in their serious adverse events section. So I read through all of those and basically came away with the conclusion of, you are more likely, literally, to be struck by lightning than have something bad happen to you because you took the COVID vaccine.
It's fine.
We
Get
can
my
talk a little
steps
bit
in.
more about adverse events while she's walking down to the microphone. It's my favorite part of drug trials. But just to give some context, she mentioned serious adverse events versus adverse events. There is a qualification that if an adverse event requires hospitalization, includes birth defects, causes death. There's 5 criteria. Do you remember the other 2? Anyway, if something's serious, it counts as a serious adverse event instead of a regular one.
I'm holding this.
I have a question about the computational chemistry that you mentioned earlier. How do you go about taking clinical trials or other types of empirical data and turn it into a model for your drug analysis?
Machine learning. And what I mean by that is if I have a series of drugs and I say this one was toxic, this one was toxic, this one wasn't, this one was, you know, a human can look at it and see, oh, there's a common motif here. This functional group, this collection of atoms in this configuration seems to be a problem. But sometimes it can be really hard to infer that from the data you have just looking at it. A, because there's too many molecules to look at, and B, because the way that we recognize things can be somewhat limited. So part of the way that we can take this data in is we can look at what things were tried, which ones worked, which ones didn't, and why they failed, and try to build models to better predict, hey, this is going to be toxic as all get out, don't even try it, versus this seemed to be very well tolerated, or this seemed to, this you know, family of drugs worked decently okay in this, but you needed to crank it up to toxic levels in the body to get it to do anything, so let's look in this chemical space and see if we can make tweaks to make it more effective, and therefore more effective at lower doses. Because a lot of what we care about in computational chemistry land is not just does it stick to the site, but how well does it stick? So can I only put a small amount in the blood and still get enough that sticks and stays on target, that it does what we need without having to pump you solid full of it?
I'm going to hold it up.
Yeah, another thing for that is how long does it persist in the body? That's a trick that they've pulled actually with Paxlovid, which is the new antiviral for COVID. Basically, it just wasn't persisting long enough to do the job, so they combined it with another drug used for a completely different indication that just lowered your ability to metabolize it. So it sticks around a little longer.
So I was just curious about the process when you take a vaccine and they make variations over time, the process for making those and getting those approved. Is it the same steps? Is it different?
So the framework that we have for that is kind of based off of the flu vaccine. And so what they have done is approved the process for developing that, or for creating that vaccine, and then have kind of left it a little blank where they can choose the different versions each year. And so what I imagine will happen with mRNA, we do not know yet, is that they will do the same thing and they will approve the process, and then the actual like sequence that they're targeting will be up for modification version after version. And it's kind of what we saw with the booster, the mixed booster. They kind of followed that same model. So we're kind of in limbo right now of how they're going to do it in the future when it's not quite an emergency anymore. But it'll be interesting to see.
On my way here, I read that they were working on the BA4, BA5 subvariant. Did they approve that while I was here in the chaos of?
Yeah,
they did.
very recently approved
It's
the
available.
cocktail.
You can book it at Walgreens and CVS in some locations.
There you go. Sign me up.
I have a follow-up.
Whoa, are you okay?
Oh, this way, over here. Yeah, sorry, I didn't think about that.
Okay. So I've got a follow-up question on computational chemistry. So once you've deduced what molecule might work, how do you go from simulation to synthesis? Like, what is your process for figuring out where to start?
So usually where you get the molecule from can come from a variety of places. It can come from virtual screening experiments. It can come from a chemist saying, I think this might work. It can come from, someone we're working with giving us some leads. So what we usually can do at that point is say, okay, I'm going to take this molecule and I'm going to make some changes to it. I'm going to functionalize it in different ways and see if I can make it a little bit more effective. Because again, I want it to not just stick to the target. I want it to stay on the target for a while and not just float off and then get eaten by the liver later on. So once you find a series of molecules that bind to your target, one of the next steps you can take is if you have a model of what you're trying to do. So for example, if I'm trying to kill a kind of cancer, I might target a kinase in that cancer, which helps it produce energy, and I want to stop that kinase, I want to stop that protein from doing its job by gumming up the works with a small molecule. So the next thing I might want to do is say, okay, if I shove the small molecule in there, can the kinase still function? And I model this by modeling the entire protein. That's one way I can go. And the other way I can go is I can just say, okay, enamine, make me these 100 compounds that I think have reasonable success of binding to this target, and send it from there to another contract research organization that basically specializes in me saying, okay, here's the type of protein I care about. Go figure out some way of making it. Here's the drugs getting shipped to you that I want to test. Tell me if these drugs stick into this target, and I can use a variety of spectroscopic techniques, so I can basically interrogate it with light to see if the molecule is stuck where I want it to stick. and figure out how much molecule I needed to get it to stick and how well it sticks on there. And then from there, I can go back and develop further. Okay, do I need to make more tweaks, or is this working pretty well and we want to get a better mechanistic understanding?
I'm a veterinary technician, and so I don't know if you guys, have a lot to do with, veterinary medicine and the drug market there. But there is a medication that's currently on the black market here in the States for what's called FIP, feline infectious peritonitis. Do you know what the process is for getting something? Because it's approved in Europe, in the EU, and I believe Canada and Mexico as well. That's where a lot of people here in the States are getting it. But with it being an injectable, and not a lot of pet parents are comfortable injecting their cat, and we legally can't do it because it's not approved. Do you guys know what the approval process for veterinary medicine in that regards is? Sorry, I know that was a lot, kind of random.
So it is a parallel but different process for veterinary medicine, and I don't think that it is one that any of us have any expertise in. I'm sorry.
Yeah, I am familiar with that one, though. I'm also a cat mom. So, yeah, I've seen, a lot of people are really excited about it because it's a very, very lethal disease, and there's finally a treatment for it. Isn't that great? And can we please get it approved already? Come on, guys.
All right. Well, I just want to sort of wrap up with some final comments, or one last question to all my panelists. Is there some sort of either or question, any drugs that you're seeing on the market that you're most excited about, or a personal drug that you think has had a really positive, huge impact on society?
Oh, yeah. PrEP. That's the big one for me. So that was first out. Gilead did a lot of the early work in HIV drugs, and now they've come out with Truvada. They came out with that about, 10, 15 years ago now. So basically, yeah, you can just take sort of low-level antivirals all the time and don't have to worry about catching HIV. It is like 99% effective in preventing HIV transmission through sex, and it's like 75% effective preventing it from IV drug use. It's an amazing invention, and it's on the World Health Organization's list of essential medications now.
I can't name the drugs that I'm excited about, but you're just going to have to trust me on this. We've got some really interesting neuroblastoma cancer treatments coming around. In the next 5 years, I think it'll probably hit market, but it's going to, childhood cancer, it's a not exciting time to be in it, but there's some really interesting treatments that are coming around the corner that I think will really change that landscape.
To be honest, I'm still pretty excited about what penicillin started for this world as a nurse.
As a neuroscientist, I'm most excited, I think, for the psychedelic therapies for anxiety, post-traumatic stress disorder, and things. Now that we're starting to get approval here for them to be done. I mean, ecstasy therapy for couples in Europe has been done for a long time, but because it was associated with a club drug here, and the government was just like, oh, the kids are using the drug in the raves and things, and we can't use them for clinical work, but now they're starting to get approved, and it's pretty amazing. One of the last trials that I read, and you guys probably know more detail about this because I get it after it's gone through all the secret stuff, but they did, it's got to be assisted therapy, and it's got to have a clinician and the dosing of the psychedelic. And they took people with post-traumatic stress disorder after war, and they had pretty intractable cases, and they intended to have 3 sessions separated by 1 month each for this psychedelic therapy, and guided by 2 therapists who were under the, who knew the patient and knew what their issues were and stuff. After the second dose of ecstasy, I think that one was ecstasy, or it might have been acid, sorry. It's been a little while since I read it. But after 2 of the 3, 2/3 of the participants no longer met the clinical diagnosis for post-traumatic stress and could not be continued in the trial because they were functionally no longer with it. So, yeah, it's pretty amazing.
And I'm just always gonna, fangirl about mRNA because it changes the game for vaccination so much. Like, it both really came in clutch for COVID, but the general idea of, oh, we have a disease and we know what part of, say, the virus we want to target, and we don't want to wait several months to go incubate it in either, eggs or Chinese hamster ovaries or what have you.
Not joking.
Yeah. And there's, it's not easy to make mRNA, but it is very straightforward once you know what you want to work with to get something spun up and get it into someone and see, is this eliciting the immune response we want, and is that immune response effective? And I think that's both going to help with a lot of diseases that we've had a really hard time figuring out vaccines for, because now we can target on a very, very tiny scale what part of, say, the virus or bacteria we want to produce and cause an immune reaction to. And it's going to really speed up vaccine development for any new diseases that come out in the future, because it's fast. It's really, really fast. And we've scaled up the technology due to the COVID pandemic situation.
Well, thank you all for coming here to this panel today. You all have been really lovely. I appreciate... Sorry. Thank you all for coming to the panel. I really appreciate you all coming here today. Again, please consider donating to Open Hand for nutritionally dense food for those who need it. And happy Dragon Con.
And if you have any questions you want to see answered by scientists on Monday at 1 p.m., we have calculations you wish we weren't doing, which is going to be order of magnitude problems like how many Jell-O shots fit in the belly of a blue whale. There's index cards and pens over there. So write your questions, throw them in the box, and maybe some of them will make it into the panel.